When we think about what keeps us alive, we usually think big: heart, lungs, brain. But what if I told you the real action is happening on a scale so small it's almost impossible to imagine? We're talking about an army of microscopic machines inside every single one of your cells, working nonstop. Let's dive into that hidden world. See, these little machines are called proteins. And honestly, they're the absolute workhorses of life itself. I mean, they do pretty much everything, from digesting your lunch to carrying oxygen through your blood to fighting off that cold you had last week. Proteins are the gears, the engines, the whole shebang that makes biology tick. But here's the really wild part. When a protein is first made, it's basically just a long, floppy string of amino acids. It's useless like that. To do its job, it has to fold itself into an incredibly specific, unbelievably complex 3D shape. Think of it like a single piece of string that needs to spontaneously tie itself into a perfect little piece of origami. So the big question is, how on earth does it find that one perfect shape out of a gazillion possibilities? And that question, well, it brings us to a huge hurdle that scientists have wrestled with for decades. It's called the protein predicament. And it's a problem so massive, it almost seems like life shouldn't be possible at all. This idea is captured perfectly in something called Levinthal's paradox. Basically, it says that a protein has so many potential ways it could fold that if it just tried them out randomly, one by one, it would take longer than the entire age of the universe to stumble upon the correct one. And that's just for one single protein. It's mind-boggling. And yet, they pull it off. In our bodies, this process doesn't take billions of years. Some proteins snap into their final shape in less than a millisecond. A millisecond. That incredible speed is the ultimate clue. It tells us this isn't a random search. No way. There has to be some kind of system, a guided pathway that funnels the protein directly to where it needs to go. And scientists have a great way of visualizing this pathway. They call it an energy funnel, just like you see here. At the top, where it's wide, you have the unfolded protein. It's high energy, chaotic, with countless possible shapes. But as it starts to fold, it's like a ball rolling down the sides of the funnel. It loses energy, its options get narrower and narrower until it finally settles at the very bottom, and it's one single, most stable and fully functional shape. It's a guided slide, not a random walk. Okay, but even with a handy energy funnel, things can still go wrong. I mean, the inside of a cell is a crazy, bustling place. So to make sure everything folds just right, the cell has its own sophisticated quality control system. It's got a team of molecular supervisors on the job. You have to appreciate just how different the environment is. Folding a protein in a clean test tube, that's like trying to do origami in a quiet, empty library. You've got all the space and time in the world. But inside a cell, that's like doing the exact same origami project, but you're in a packed subway car during rush hour. It's insanely crowded. In that chaos, a new unfolded protein is way more likely to just bump into something else and stick to it, forming a useless, mangled clump. So the cell needs help, and that help comes from an amazing team of specialized proteins called molecular chaperones. You can think of them as the cell's personal folding coaches, or maybe even its bodyguards. Their whole job is to guide new proteins, keep them out of trouble, and make sure they fold correctly. Now, this is not a one-size-fits-all kind of operation. The cell has an entire toolkit of different chaperones, each one a specialist for a different job. Altogether, they work to maintain something called proteostasis, which is just a fancy word for keeping all the proteins in the cell healthy, balanced, and functional. And just look at the variety in this toolkit. It's pretty amazing. You've got these big barrel-shaped guys called HSP60s, these cool ring-like HSP100s, and then the more flexible HSP90s and HSP70s. Every family has a unique structure because they all have different, specific jobs to do. They're a highly coordinated team. And their job description? It's huge. As you can see here, they do everything. They shield brand new proteins right as they're being born. They can help unfold proteins that need to be moved or recycled. And they can even perform rescue missions, pulling valuable proteins out of those dangerous clumps. They really are the masters of protein management. So how do they actually do all this? It's one thing to talk about it, but let's see a chaperone in action. We're going to zoom in on one of the most important and versatile members of the team, the HSP70 machine. HSP70 runs on a simple but incredibly powerful four-step cycle. First, it spots and grabs onto a little segment of an unfolded protein.
Second, fueled by energy, it clamps down hard, holding that protein tight so it can't get into trouble. Third, it just holds on for a moment, protecting it from that chaotic subway car environment. And finally, it lets go, giving the protein a fresh, clean shot at folding the right way. And if it fails again, no problem. HSP70 just grabs it and starts the cycle all over again.
And you can see that clamping mechanism right here. On the left, HSP70 is in its open state, ready to grab a protein. Then on the right, after it uses a bit of energy, you can see how it physically snaps shut into a closed state, creating this super secure hold. It is, quite literally, a molecular machine with moving parts. Just amazing. This whole system is so elegant, so precise, and it is absolutely essential for us to live. Because when this system fails, when good proteins go bad, the consequences of that misfolding can be truly devastating. So let's go back to our energy funnel for a second, because it turns out there's a dangerous side path. Instead of sliding smoothly down to the bottom, a protein can get stuck, falling into a different energy pit. And this path doesn't lead to a working protein. It leads to aggregation, where these misfolded proteins all stick together into toxic clumps. And this isn't some abstract problem in a biology textbook. Protein misfolding is at the absolute heart of some of the most difficult and heartbreaking diseases that we face. These toxic clumps, or aggregates, are what build up in the brain in neurodegenerative diseases like Alzheimer's and Parkinson's. It's the cause of prion diseases like Creutzfeldt-Jakob. It's even linked to many cancers, when critical proteins that are supposed to protect us fail to fold correctly. In every single one of these diseases, the basic problem is the same. Proteins fail to find their correct shape. They start clumping together, and these toxic aggregates start piling up in our cells, causing immense damage and ultimately leading to disease.
So at the end of the day, we depend on this incredible network of chaperone proteins every single second. What we're now learning is that this network can get overwhelmed by stress, and it seems to get weaker as we get older. And that leaves us with a really powerful and hopeful question for the future of medicine. If we could figure out how to boost and strengthen our body's own chaperone network, could we one day stop these devastating diseases before they even get a chance to start?